Welcome back in another lesson of virology and microbiology. Just as I promised, it will be a follow-up for my last movie about the virus called SARS-CoV-2 and the not so nice influence of its spike proteins on the general state of health of infected person. So, if you just got in and wish to enjoy a somewhat smooth ride with the rest of the class, you should probably get yourself accustomed with the things which were discussed in my previous lesson, or the amount of new information might get too big to be processed by someone who doesn't deal with molecular biology in his everyday life. You have been warned. You might remember that the science presented in the last movie could appear as quite controversial to some random commoners, as it didn't go too well with the generally approved narrative. Well, science doesn't care about the mental comfort of the public, and so with each presented publication, things started to become pretty unsettling and quite worrisome. And don't expect that this movie will give you some relief. If anything, it will most likely lead your mind even deeper into the abyss of fear and despair. What you saw last time was just the general outlook on the gloomy landscape up ahead. Now it's time for us to confront all speculations with the unforgiving life. But before we start dealing with hard scientific data, let me first provide you with some small theoretical background. To be more specific, I need to speak about the ways in which the host body is able to deal with the disease. As you might know, the main biological defense against the infection is provided by our natural immune system. But don't worry, I won't discuss here all the details of this quite sophisticated mechanism. For now, let me just say that in order to fight an ongoing infection, immune system is producing enzymes called antibodies. There are, however, some problems with such protection. Each kind of antibody has a very specific specialization of its task, and in order to have maximal efficiency, chemical chain of a given antibody has to perfectly match the targeted viral protein. In shortcut, every single protein of the virus is being targeted by its own specific antibody that won't work on any other viral protein. Also keep in mind that immune system needs some time to figure out what type of antibodies it has to produce in order to completely destroy the infecting virus. In the case of COVID-19, it takes around 2 weeks for the innate immune system to develop and produce proper antibodies. During this period of time, SARS-CoV-2 virus inside infected body can replicate and spread to other people without any restriction. There are, however, a couple different ways in which we can help our immune system to deal with this issue. One of such ways is to provide the infected person a chemical substance that works as an inhibitor for the process of spike proteins binding to ACE2 receptors on the host cell membrane. Such treatment doesn't kill the existing viruses, but doesn't allow them to replicate further. So in order for it to work, the inhibitor has to be introduced in the early stage of infection, first couple days. Here is an animation which shows how an inhibitor stops the SARS-CoV-2 from entering a host cell. Of course, we know already about a couple of such inhibitors. According to some scientists, one of the most potent ones is known as ivermectin, used as human and animal dewormer. However, for some reason, which I won't discuss here, it is being prohibited by some health agencies in the early treatment of COVID-19. Another way of dealing with the delayed response of the immune system is to use the antibodies extracted from the blood of people or animals that already recovered from COVID-19. As you might guess, such treatment is quite efficient and works in all of the stages of SARS-CoV-2 infection. In shortcut, it works similarly to antibiotics for bacterial infection, only it cleans you up from a particular protein characteristic to a particular virus. If you belong to the smarter part of my audience, you might ask right now, hey, then why aren't we using the antibody treatment as their primary way of dealing with COVID-19? Yes, indeed. Why aren't we? Monoclonal antibodies are being successfully used in the treatment of COVID-19 since early 2020, and they seem to be very efficient. Although there are some countries and states where this kind of treatment is being provided by health agencies to all citizens already for some time, in 98% of the globe, it's an unofficial pay-to-win option that will cost you at least $400 for 1 milligram dose. Only in last month, some countries in Europe started to rather slowly open the door to the official approval of the antibody therapy for COVID-19 and mentioned about the refundation of such treatment. But somehow no one seems to be especially in hurry. Of course, if you can afford it, there are no problems for you to order it on the internet. Just look at the sites visible in Google Ads. Yeah, well, it might make some of you wonder if it isn't just another part of some greater conspiracy and or a smart way of making couple extremely wealthy people more wealthy. But who am I to know such things? And finally, there is also the third free-to-play option, known as vaccination. The general idea is quite simple. It's about exposing the immune system of a healthy person to a harmless part or version of a virus so it can learn the proper response in case of actual infection in the future. And now I will probably disappoint some of you, but I have to state it clearly. Yes, in general, vaccines do work as they supposed to, and yes, they are generally a safe and effective method of making uninfected people immune to some of the most severe diseases. And I say this as someone who was vaccinated many times before. With that being said, let me now explain what's the main difference between the classic vaccines that were being used up until now and those which are being currently used in the race of immunization for SARS-CoV-2. Difference is here quite significant. Up until now, vaccines were based on weakened or disarmed versions of the wild viruses that made them unable to replicate and cause the actual illness as the result of that process. In case of COVID-19, all of the four main brands of vaccines that are currently allowed for the public use, Pfizer, Moderna, AstraZeneca, and Johnson & Johnson, are based on a completely different technology that wasn't used before. In shortcut, those vaccines contain small fragments of genetic code which instructs the host body how to produce the proteins that's supposed to be targeted by the immune system. What makes the most important difference between the anti-COVID vaccines is the way in which this code is being delivered to the host cells and utilized. But before I will go any further, I need to make something clear to all of you. I don't have no medical education and or experience, except being in hospital as a patient. If you want to hear a professional advice regarding safety of your health, don't ask me. My private opinion about presented subjects doesn't matter in any possible way. In shortcut, I have no valid right to tell you what you should or shouldn't do with your body. I leave any decision regarding your health to you, or in some cases to your country state. Ugh. As I said couple times already, I am not in the position to present my private opinion about the safety and or effectiveness of available COVID-19 vaccines as reliable and or valuable to anyone. I can however make the promise that information presented in this movie will be fully consistent with the generally accepted science, so you might be able to discuss it with someone who has proper credentials and make your own opinion by yourself. Let's then begin from an article called Targets and Strategies for Vaccine Development Against SARS-CoV-2, which should make a nice frame for the discussed subject. Here are the highlights of the main subjects that are being discussed in this article, and I will go through them in a second. However, I want to begin from a statement that can be found in the conclusions at the very end of this publication. And here is that statement. Vaccine alone cannot combat the pandemic. A statement that the big pharma corporations nor most of government officials won't likely agree with, as the main strategy which is being currently presented to us is to make the entire human population vaccinated. Well, this and forcing on us seasonal lockdowns. However, since the worldwide vaccination program seems to be currently the main way in which the people in power want to combat the still ongoing pandemic, let's see what should be the features of a perfect vaccine, one that should become the goal to achieve for all vaccine producers. Such a perfect vaccine should be safe, be highly effective, be free from toxicity and adverse effects, give long-term protection, and have high thermal stability. As I mentioned many times before, my opinion regarding this subject doesn't matter, but if it would, in order to make a vaccine which would fit in most of those criteria, I would base it on the entire SARS-CoV-2 virus with exclusion of those parts of it that allow it to enter the uninfected host cells. That means the spike protein as a whole or just the RBD on top of the spike. Without it, the virus should be completely harmless, as it wouldn't be able to fuse with the host cell membrane and initiate the script of replication. At the same time, such disarmed virus, which allow the immune system to learn how to produce the antibodies against the M, N, and E structural proteins, together with the non-structural ones, providing us with the necessary defense mechanism against the actual virus, without the danger of the vaccine causing COVID-19, which is generally the effect of massive replication of the virus by the infected cells. Such immunization should be also a long-lasting one, since mutation don't affect the whole virus at the same level, and even if dealing with a mutated variant of the original form, immune system should be able to recognize at least one of the viral proteins, giving us enough protection to overcome the possible infection. But let's say that for some reason my vaccine has to target only one of the viral proteins, just like in case of the available vaccines. Which one would I then choose? Well, it would be the one which is supposed to be harmless to healthy cells, one which is abundant and which is crucial to the proper functionality of the virus as a whole, while maintaining a relatively high mutational stability. Having all of this in mind, my choice would be the viral membrane protein. Of course, who am I to decide about such things? It will be much better to leave such decision to a computer simulation, which is much less prone to make mistakes in the calculations. Yeah, well. It seems, however, that people responsible for making actual decision when it comes to specified targets for new vaccines have a completely different perspective on the process of immunization than I or the AI has. While my own perfect vaccine would be based on the entire SARS-CoV-2 virus except its spike proteins that make it dangerous to human body, it's actually this spike protein that is being targeted by almost all major vaccine producers. Currently, out of 47 vaccine candidates under clinical trials, around 45 are spike protein based. What all the major vaccine brands have in common is that they all make previously healthy cells of vaccinated person to produce the spike proteins by their own, similarly to the process of actual viral replication. This decision was based on the spike protein being A. Necessary for the virus to enter the uninfected host cell. Without the spikes, the virus would be mostly harmless. And B. Capable of inducing a strong neutralizing immune response. However, there are also quite a lot of differences between each one of the approved vaccine brands. One of such difference is about different technologies used in different vaccines to deliver the genetic instruction of S protein expression by the compromised cells of vaccinated person. Here are the four major vaccines available to the public and the technology that is being used by each of them to teach healthy cells how to create the spikes without any actual contact with the SARS-CoV-2 virus. As you can see, vaccines from Pfizer and Moderna are based on technology that utilize mRNA, while those made by AstraZeneca and Johnson & Johnson are using virus-based vectors to deliver the genetic code to cells of vaccinated person. Another way in which the currently available vaccines differ between each other is defined by the type of spike protein that is being utilized by each of them. As for the four major vaccine brands that are approved by most of public health agencies, only the one produced by AstraZeneca uses a full-length spike protein in its natural form that doesn't differ too much from spikes attached to the actual virus. As for the Pfizer, Moderna, and Johnson & Johnson anti-COVID vaccines, they utilize spikes with coupled distinct artificial modifications. If you saw my previous movie, you should already know a bit about the general structure of SARS-CoV-2 spike protein, its conformational characteristics, and the processes which is involved with. But for those who don't know anything about this subject, spike protein allows SARS-CoV-2 to infect host cells by binding to ACE2 enzymes on the surface of couple specific types of cells that can be found in most of major organs of a healthy body. The entire process in which the spike protein binds to ACE2, leading to fusion of membranes and subsequent viral infection, might be somewhat complicated for those who don't know anything about it. I warned you about this in the beginning of this movie, and I really don't like to waste the time by explaining once again things that I've already explained not so long ago. However, since this part will be crucial for many other subjects that will be discussed in this movie, I will now show you the most important details of that process. Here is a fragment of a movie that should tell you much more than my awkward verbal explanations. In shortcut, before the fusion with a host cell, spike proteins of SARS-CoV-2 remain in a pre-fusion state, which allows them to bind to ACE2 proteins using the receptor binding domain, RBD, on top of each spike. When it happens, the binded spike goes through a couple different processes that change its structural composition as it enters the post-fusion state, in which the spikes can't bind to ACE2 anymore but become responsible for the fusion of membranes. I'm telling you all of this because both mRNA-based vaccines, Pfizer and Moderna, encode spike proteins that are artificially modified to remain permanently locked in the pre-fusion state, what's supposed to significantly reduce their ability of binding cells together, what can lead to the formation of blood clots. Spikes utilized by the Johnson & Johnson's vaccine contain a different modification which, although prevents the cleavage of spike's S1 subunits, still allows it to get into the post-fusion state. What matters, however, at most is the fact that in all of the available anti-COVID-19 vaccines, the spike proteins that are being utilized by them are fully capable of binding to ACE2. And as you'll see soon, this information is the most crucial one when it's speaking about the toxicity of spike proteins. Having all this in mind, let's now look closer at some properties of the spike protein which should be taken into account when it comes to designing a properly working vaccine. Let's begin from the mutational stability. To be a good target for a vaccine, spike protein shouldn't be affected too much by mutations. Quickly mutating proteins will allow immune escape, rendering the available vaccine pretty much useless. Well, after 1 year of the ongoing epidemic, it was possible to say that spike protein, together with the nucleocapsid protein, are the ones with the highest level of mutational variability. Of course, it makes sense for someone who wants to be assured that there will be a constant demand for new upgraded vaccines each time when a new variant of the virus will emerge. Of course, just as before, I don't claim that it's any kind of conspiracy. It's for you to decide. However, the possibility of immune escape is just one issue. High rate of mutations might also lead to the effect known as ADE, antibody-dependent enhancement, in which the immune system of vaccinated person actually helps the virus to enter uninfected cells, leading to the increase of illness severity. Be sure that I will come back to this subject later in this movie. But now, better get a hold of yourself, because things will get pretty spiky, as we will look at the general safety of spike protein for a healthy human body. Toxicity of the spike protein was the main subject of my previous movie, so if you want to learn more details, I suggest you to watch it. But for now, let me give you the main highlights of this subject. Let's begin from the fact that the spike protein doesn't need the rest of virus to cause serious damage to healthy tissue. Here is the lungs, and cause symptoms of COVID-19 even without the process of viral replication. I won't here go deeper into this process, but what the spike protein does is to initiate the apoptosis, a literal algorithm of self-destruction of healthy and uninfected cells, just by binding to ACE2 and disrupting its natural regulatory function. This is exactly why I told you to remember about spike proteins in the vaccines being able of ACE2 binding. This particular property of the spike protein makes it a full-blown cytotoxin. However, the spike protein not only kills previously healthy cells, but it also induces an inflammatory response of the immune system, what can lead to a cytokine storm and hyperinflammation, which is a potentially deadly condition associated with the so-called long COVID. Another interesting property of the spike protein is its ability to pass through and disrupt the blood-brain barrier, BBB, what gives it the title of a potentially deadly neurotoxin. And finally, it has also the potential to alter the DNA of a healthy person and disrupt its natural damage repair mechanism, what was proved by in vitro studies. I'm sure that there might be more ways in which the spikes of SARS-CoV-2 can mess up the human body, but as for now, it's probably more than enough to give you the general idea regarding the credential of spike protein when it comes to being a deadly toxin. Of course, all those papers discuss the toxicity of spike proteins in their natural form and attached to the SARS-CoV-2 virus, not to be mistaken with those spike proteins that are being produced by the cell of a vaccinated person. I mean, considering all the potentially harmful properties of those spikes, vaccine producer did probably everything to be ensured that the spike proteins used in their vaccines are completely harmless to vaccinated people, right? Right? Well, according to experimental and practical science, SARS-CoV-2 spike protein is literally a deadly toxin because, among many other properties, it causes destabilization of regulatory functionality of the ACE2 enzyme, what leads directly to subsequent death of healthy and uninfected cells. And as I stated couple times in this movie, spike proteins used in the four major brands of approved anti-COVID-19 vaccines are fully capable of doing it, since all of them can bind to ACE2. But of course, as I stated multiple times already, I am not in the position to tell you if a medical procedure is safe or not. It's for the patient to decide about it. All I do is to make sure that the patient know about the possible danger of a treatment, which the clinical and experimental science knows about at this particular time. And I think that up until now I provided enough credible and reliable sources to prove quite clearly that the SARS-CoV-2 spike protein is indeed a pretty dangerous toxin, capable of causing serious damage to our vital organs and or death. And now I'll give the voice to all those experts who claim that the spike proteins utilized in the major brands of approved anti-COVID vaccines are actually safe to the vaccinated person. I went through multiple fact-checking sites to see what some of their experts tell us about toxicity of the spike protein, and now I will dissect here their statements. Let's begin from those two marked statements made by Mr. William Matchett and Dr. Dan Cole, according to which spike protein is not a toxin. My question then is, what is the scientific base for such claim? Because I can't see any. It seems that them being experts supposed to be enough when it comes to evidences. Well, I'm terribly sorry to say it, but there is enough scientific evidences of the spike protein being a toxin to tell that claims of Mr. Matchett and Dr. Cole are completely wrong. Obviously, they aren't particularly interested in learning about recent studies or in reading papers published in medical and scientific journals. Or maybe spreading medical disinformation is their intention. Of course, I invite anyone who doesn't agree with my fact check, including experts, to disprove and or invalidate the sources which are presented here, link in description box. And those who would like to discuss my interpretation of the sources can always find me without any problem in the comment section in any of my movies. But something tells me that there won't be too many experts willing to speak with me. Okay, so now let's try to discuss the statement at a higher level of difficulty. One that claims that there are no evidences of the spike proteins from the anti-COVID vaccines being toxic or in any way harmful to the body of a vaccinated person. Well, first of all, there is actually quite a lot of verified and reliable sources of vaccine-induced injuries, including ones that were made post-mortem, and I will present some of them soon in this movie. But let's assume for now that there really aren't no such studies, and that such claims are actually true. What a strange time we're living in. Time when in order to turn a random statement into a 100% confirmed scientific fact, all you need is some guy labeled as an expert who will state that he personally didn't see any evidence of that random statement being incorrect. Cool. So here's my claim. T-Rex would make a perfect household pet, as he would never eat or harm in any way a human. And now I just need to call some random expert in the field of paleontology and ask him/her if he has seen any evidence of people being eaten by a T-Rex. And voila! My claim is now a fully established scientific fact. Obviously, this is how you make science these days. It's one thing, however, to state if some evidences were or weren't seen personally by a random person, expert or not, and it's a completely different thing to give the absolute answer to all possible questions about the actual existence of those evidences. I know that it might be a shock to some of the experts, but the fact of them seeing or not seeing evidences doesn't tell us nothing about those evidences being real or not. In this particular case, there is direct proof of serious injuries caused by the COVID vaccines, even if it wasn't seen by any of the reference experts. And soon such proof will be presented to you in this movie. Okay, so now let's look at some claims that have an actual scientific base. In fact, I've already fact-checked them in my previous movie, but since they are rather crucial for the discussed subject, I will fact-check them for you once again. As we can read on multiple fact-checking sites, spike proteins utilized by the vaccines for COVID-19 are not the same as the spike proteins in the actual SARS-CoV-2 virus, and because of those differences, they are completely and absolutely safe to the vaccinated person. Those are the three main differences between the spikes used in vaccines and those which are used by the virus that are being pointed out by the fact-checkers. 1. Spikes used in vaccines aren't attached to the virus, so they can't cause the illness called COVID-19. But of course, I've already presented you a paper which proves rather clearly that spike proteins alone are more than capable of causing some rather serious damage to a healthy tissue. 2. Spikes used in currently available COVID-19 vaccines are genetically modified to remain locked in the pre-fusion state, conformation. First of all, such modified spike proteins are being used only by two brands of anti-COVID vaccines. Spikes utilized by AstraZeneca and Johnson & Johnson can turn into the post-fusion state. And second of all, you saw already a paper where inventors of technology that locks spike proteins in pre-fusion state stated themselves that those spikes can bind to ACE2 despite this modification, and it has nothing to do with syncytium formation. Shortly, what you see here is a first degree of scientific and medical disinformation. Spikes used by some vaccines are genetically modified to remain anchored to the host cell membrane. But even if they are, it still doesn't stop them from disrupting the physiological function of ACE2 in surrounding cells, causing their death. Besides that, there are at least couple studies proving that the spikes produced by human cells due to vaccination don't stay only in the general area of intramuscular injection, but they are indeed circulating in the body for some time. I will show you the sources pretty soon. But I have to admit that the efforts of all those experts trying to convince the public about the general safetiness of anti-COVID vaccines are nothing less but adorable. Here, for example, we can see how one expert is at least trying to stick with the established scientific facts, what leaves her with no other choice than to admit that the spike protein is indeed a toxin, despite multiple other experts stating otherwise. So then she tries to prove that in the difference to natural infection, spike proteins used in vaccines are not toxic because they are anchored to cell membranes, what stops them from circulating freely in the body of vaccinated person and destroying tissue in his vital organs. So they aren't toxins until they stay in the muscle at the point of injection, but become toxic in any other part of the body. It seems as well that in some cases those fact-checking sites aren't just contradicting other fact-checking sites, but they even seem to directly contradict themselves. Here, for example, you can see as a big bold-typed statement about a complete lack of evidences proving that the vaccine-generated spike proteins can be lingering in the bloodstream is being quickly disproved in the fourth paragraph under that statement. Dear Mr. Fact-Checker, may I ask why didn't you just state that the spike protein can indeed be detected in the bloodstream of a vaccinated person up to a couple of weeks after the vaccination, but the observed amount are actually pretty tiny? This is all you had to do to be somewhat consistent with facts. It seems, however, that not everything from those fact-checking articles is a full-blown scientific and medical disinformation. I've managed to find one fragment which can be considered as partially true, Despite the supposed total lack of evidences, this particular fact-checker admits the possibility of spike proteins being released into the bloodstream when the spike-producing cells die. However, rather than usual day-to-day cell death, it's much more likely caused by the spike-induced apoptosis and subsequent autophagy of the genetically compromised spike-expressing cells. Still, it seems like a kind of progress. Of course, according to him and some other fact-checking specialists, observed quantities of spike proteins in the blood of vaccinated people is far too low for them to be considered toxins. Funny, because I thought that substances are just considered toxins or they aren't, no matter of their quantities. Does the rattlesnake venom stop being toxin if there is not enough of it in the blood of a bitten person to cause a serious injury to his body or kill him? I would have to ask some experts. But I don't think that this particular fact-checker is capable of making those things somewhat clear. As someone who produced a Twitter infographic showing the harmless nature of vaccine-generated spikes is for him an expert in the field of molecular biology. Well, in such case, we can also refer to a janitor in the art gallery as to a grandmaster of art. However, in other fact-checking articles, it doesn't get any better. Here, for example, we can learn that since the spikes are anchored to the host cell membranes and the vaccine is delivered to the muscle tissue on your shoulder, they can't be circulating in the bloodstream as they will stay around the injection point. And at the first sight, it might seem that this is exactly what is being confirmed in this article. Vaccine-generated spike proteins remain stuck to the cell surface around the injection site and do not travel to other parts of the body via the bloodstream. But a teeny tiny portion of those spikes, which actually does go beyond the injection point and does get into the bloodstream, is being swept into the liver and lymph nodes where the spike proteins are being quickly destroyed by enzymes. Sadly, I couldn't find any experimentally verified information regarding the quantity of spike protein at which it turns into a toxic for the human body. So instead discussing results of the study which is mentioned in those fact-checking articles, I will show you a much better source instead. Here's a paper which can give us a pretty reliable answer at least to the question about the circulation of vaccine-generated spike proteins in the body and their persistence in plasma of a vaccinated person. Link is in the description box. This particular study concentrates mostly on SARS-CoV-2 spike proteins that are being expressed by circulating exosomes, cells that allow communication between distant parts of the body. Authors were able to identify the spikes attached to circulating exosomes using a couple different techniques, including images from an electron microscope. To the left, you can see the results. But here is the most important part. According to the observed results, here graphs B, D, and E, spike proteins were detected in circulating exosomes soon after vaccination with the Pfizer mRNA vaccine, with the density peaking around 14 days after receiving the second dose, and remained at detectable levels even 4 months after the shot. I won't go here deeper into the details, but if you're interested in the subject, simply Google out circulating exosomes, and you will see that it's yet another mechanism that is associated with multiple pathological disorders like cancers, strokes, or cardiovascular diseases. Authors extracted then the spike-expressing exosomes from the plasma of vaccinated people and injected them into healthy and unvaccinated animals, mice. It was then observed that both the vaccine and the exosomes induced a similar immunological response in the test subjects. Mainly due to both injections, there was a significant increase in the number of T cells producing interferon gamma and tumor necrosis alpha cytokines. Normally, I wouldn't speak about this part of study, but just so happens that only a couple days ago I found a fresh study straight from the bakery which discusses the involvement of those two cytokines in the severity of COVID-19. I think that the title of this paper should tell you everything what you need to know about the influence of synergism in TNF-alpha and IFN-gamma cytokines on the severity and mortality of COVID-19. In shortcut, combination of those two particular cytokines significantly induces inflammatory cell death, tissue damage, and mortality associated with infection of SARS-CoV-2 virus. Not exactly the kind of immune response which is preferred for the survival of infected person. But if you want to have a better understanding how strong is the effect which the combination of TNF-alpha and IFN-gamma cytokines has on the number of cells that are being killed due to inflammation induced by autoimmune response to SARS-CoV-2 infection, just look at those graphs, which show the percentage of cell death in response to different combinations of different cytokines. Of course, it's easy to see that the combination of TNF-alpha and IFN-gamma results in the number of dying cells which is many times higher compared to any other known combination of cytokines. And now keep in mind that this kind of immune response to SARS-CoV-2 infection is the one that the body learns thanks to vaccination. Okay, that's it for the intermission. Let's go back to the fact checkers and their fact contradicting statements. I want to quickly discuss the second publication that is being mentioned by multiple fact-checking sources. It's an experimental study in which the scientists encoded a tracking marker, luciferase, as mRNA enclosed in lipid nanoparticles, mechanism that's being used in Pfizer and Moderna vaccines, and delivered it via intramuscular injection to rats, although some say that these were hamsters. The general idea of this study was to observe the biodistribution and expression of injected mRNA in the body of vaccinated person. But before I'll proceed with this subject, I'll have to say goodbye to the fact-checker from Medium.com. You see, I'm trying to avoid being banned by the YouTube censorship committee by being fully consistent with the generally accepted science. So I can't refer to a source which is obviously spreading scientific and medical disinformation, like the one presented in this article. Hard to say if it's because he tried here to think by himself and it didn't go too well, or it's because he's referring to some low-grade expert of questionable quality and minimal understanding of the subject. What matters is that with just one short statement, he managed to completely discredit himself. Actually, it's not that hard to guess that in fact there is a significant issue with the fact that this experiment is supposed to represent the biodistribution and concentration of luciferase created from the injected mRNA, and not the biodistribution and concentration of spike proteins. To be honest, the differences are here so huge that it's actually hard to point out even a single property that would be shared by both the spike proteins and the nanocapsules with luciferase encoded in mRNA. For now, let's just say that the mRNA containing lipid nanoparticles are capable of initiating expression of the proteins which they encode in the cells that were compromised by them, but they are not capable of binding to ACE2 and or initiating apoptosis of cells. Spike proteins are on the other hand highly cytotoxic and destructive towards all kinds of cells expressing the ACE2 enzyme, but they are unable to replicate by themselves. At least they shouldn't be, and you better pray for this to be true. I don't like to be considered a snitch, so maybe this time I won't report him to the upper echelon. Let's just assume that he's spreading disinformation due to nothing but sheer ignorance, and not due to some dark and evil intentions. Okay, now back to the main subject. As you saw a couple minutes ago, in the established approved fact-checking articles, you could find the information about 99% of the vaccine-induced spike proteins remaining in the muscle at the injection site, and the 1% that goes beyond it being quickly destroyed by enzymes in the liver. Of course, I proved you already that it's not true, as spike proteins use the circulating exosomes as their vehicles to travel freely all around the body of vaccinated person for many months following the injection. But it still doesn't end here. You see, the Japanese study referenced in couple fact-checking articles allows us to directly verify the claims about 99% of mRNA from the vaccines remaining in the muscle at injection site. So let's do exactly that right now. And surprise, surprise, it turns out that even if most of intramuscularly injected mRNA containing lipid nanocapsules remained indeed in the muscle close to the point of injection, there was some measurable part of the load that got transported into many different parts of the body, and this part was far greater than the supposed 1%. Still, you need to remember that those studies are based on mRNA which encodes luciferase instead of spike proteins, and that scientists used mice instead of people as their test subjects. But of course, I couldn't find any information on how those differences might affect the final results of the biodistribution study. For now, it's just yet another big unknown we have to live with. There is also one more thing that seems to be completely ignored by both sides of the discussion, And it's not that this information is confidential or hard to find. I found it in the article called The Spike Protein and the Pfizer Vaccine, which you can read on New Zealand's EPA site. As we can learn from this article, Pfizer mRNA vaccine has couple extra features, which should be probably included in the biodistribution study, but most likely aren't. What are those extra features, you ask? Well, since the toxicity of spike protein is already an established scientific fact, and everything what's supposed to make the anti-COVID vaccine non-toxic is the intramuscular distribution and low quantity of produced spikes, according to a couple of the presented fact-checking articles, one might probably guess that those extra features were added to decrease the amount of produced toxin and/or stimulate the immune response so the harmful proteins can be destroyed before they manage to cause some serious harm to the body. Yeah, well, not really. In fact, those extra features of Pfizer anti-COVID vaccine were designed to achieve exactly opposite effect. Not only they suppress the innate immune response of vaccinated person, but they also increase the amount and production rate of spike protein expressed by the vaccine compromised cells. Sadly, I couldn't find any data which would allow me to say how the quantity of spike proteins produced by cells due to vaccination compares to the quantity produced due to natural infection with SARS-CoV-2. Please let me know if you know the answer. In practice, it means that even a very low concentration of the mRNA containing lipid nanocapsules from Pfizer vaccine can potentially trigger a local flood of highly toxic spike proteins produced by cells of infected organs. And keep in mind that some parts of body are made of cells that are rather delicate in nature, like the network of neurons in a properly functioning brain. So even a relatively small concentration of spikes in the brain tissue might potentially lead to traumatic and life-threatening injuries in the nervous system. Having all this in mind, let's now look closer at the observed results. Data from the table is given here in form of a graph. As you can see, in 48 hours since the vaccination, vast majority of injected mRNA remained in the muscle tissue near the point of administration, what is represented here by the dark gray line. There is, however, something about this data what probably should be pointed out by someone, but isn't. Notice that the amount of active mRNA in the muscle at injection site reached its peak just 1 hour after the vaccination and then started to gradually decrease. Just 48 hours later, its amount was already 50% lower compared to the peak value. However, if we look closer at the amounts of active mRNA in other parts of the body, we'll get a completely different story. Until 4 hours since the injection, the amount of expressed luciferase was indeed very tiny everywhere in the body except the liver. But then, in multiple different organs, in ovaries and prostate among many others, the amounts began to gradually increase. And 48 hours later, they didn't appear getting anywhere close to their peak values. If you would like to know how long the expression of mRNA encoded proteins might be taking place in different body parts and what amounts it can reach at its peak, congratulations! Your brain functions seem to exceed those which are characteristic to low-tier multicellular life forms, like annelids. And if you ask those questions before the ingestion of substance in question, you might be even smarter than Anastasia. Huh? You are still waiting for some actual answer? Lol, what a bunch of losers. Go home and rest assured that you'll be the first ones to be informed if someone from the office will actually try to address this problem in any way. Of course, you can also try calling us at any time. Just don't expect that someone will answer your call. Good luck.